And now we are ready to start again. As you can see from the title, the next talk will be about interactions in proteomics and networks. We had also, as you remember, we had several talks about interaction between proteins, like, for example, Albert Hecht's talk, also Paola Piccotti's yesterday. So there is a lot of work to do regarding interaction, and also is a very interesting topic in proteomics. If you remember, you already seen this image from Bella's presentation, but this time now we added something more. We have the interactome here at the top. As you can see, it's bigger than, it's like a reverse pyramid, so it's bigger than the others. So this means that there is a lot of stuff about interactome. We have interactions between RNA and DNA. Interactome studies all the interactions between these are the omics. So we have interaction between RNA and DNA. We have interaction between protein and DNA, protein and RNA, RNA and others RNA. But for today's talk, we will focus only on interactions between protein and other proteins. One issue in interactomics is the problem that, as you can see from this very simple image, imagine that this protein is, for example, an antibody. And we have here two other proteins that are binding this protein where they are where they are supposed to be, and also they are the correct proteins. So we will call these specific binders. Well, the problem is that many times we can have also proteins that are not supposed to be part of this complex, and these are unspecific binders. So a big issue in interactomics is to be sure that what you're selecting, what you're analyzing, are only proteins that are supposed to form complexes or to be bound together, and to wash away all these possible contaminants that you will have, all the possible specific, unspecific binders. How is this done? Well, an example here, probably you know better than I do this, because these are wet lab techniques. This, for example, is tandem purification. We have here our protein of interest, the one that we want to study, that is bound to another protein A that will be used as like a bait. And we have the calcium binding protein here. Then we will bind this protein with a specific binder here, probably an antibody or something like that. But here in this moment, we may have something else like around here or here that is not supposed to be bound to that antibody. So we don't want to analyze also that. So we cut away this part. Then we select only the calcium binding protein here. We do another wash. So we remove other potential contaminants. And then we remove the calcium, and what we should get at the end is just our protein of interest, so the protein that was part of the complex that we wanted to study. There is also another way to do so that uses protein quantification. This is called affinity enrichment mass spectrometry. As you can see from this very simple image, what we have here, we have an antibody. Then we put into our sample a bait that we know will bind other proteins, will form a complex with the proteins that we are interested in, that we are actually looking for. And if then we do quantification, we will get here the spectra of all the proteins that were attached to the bait, and also the gray one, the unspecific binder that just was attached here in a part of our antibody that we're not interested in. But then we do the second experiment without the bait, and after washing away all the proteins inside our sample, the only thing that should remain similar to the previous experiment are the unspecific binders, because we expect this protein not to be bounded anymore to our antibody because the bait was not present there. So then we calculate here quantification, we do like a volcano plot, and we don't expect to find any significance in the baits, in the unspecific binders, and then we expect instead to find significance in the proteins that form this complex. So I'm going to show you now an experiment that was done, is a bit old right now because it was done in 2015, but it was the first time they used they use this technique to quantify complexes. And it's particularly interesting also because you're going then, in the practical part of this talk, you're going to repeat the same experiment that Kielhauer et al. Did in 2015. So maybe you will understand better than I do also this part. This is just how they did the experiment. They had yeast cells with all the proteins inside that they wanted to study. Then they selected specific baits for each experiment. They did several replicates with several type of baits. And they then selected through antibodies the bait that, as you can see from this image, we have like here the antibody, B is bait, then these are the specific binders, and these U here are the unspecific binders. So they watched everything, they took everything that was here in this complex, they digested, then they get the peptides, and they proceed with the mass spectrometry analysis. So then here you can see MSMS scans. And lastly, what they get was a matrix, where we will have here baits and here praise. Baits are those that were used to select the complexes, and praise are those proteins that end up to be differentially expressed, not differentially expressed, but let's say they were like differentially expressed, but because we just select them in the experiment with the bait, and without the bait, we are not going to have them. So these are the praise. We do a matrix, and then we expect to find which complexes, which proteins interact with which others. Here, if you're interested, there are just a couple of information regarding the experiment, the wet lab setup that they did. So they insert this part here with a GFP protein to be attached to the antibody. They use these clone collections of yeast GFP. And then there were 30 different baits used, and for each one they replicate the experiment 4 times. Then we have this one, just to let you know, they also insert another GFP on the histone gene, just to be the control. But this control group was without the bait. I mean, with the bait only for this one, but they were not analyzing the other baits that they were interested in. So let's go to the results that they get. As I said, how you represent these results, you do a volcano plot, like what was shown yesterday. As you can see here, we have our control group with the histone only differentially expressed. And then here we have other 2, for example, in this case was used a particular bait. Now I don't remember which one exactly. And this bait then formed a complex with these 3 actually proteins that we can find here again. But what is interesting here? This is a very nice result because it's something that they implemented in their work. So instead of using, for example, let's look at this volcano plot here. Here, well, as you can see here, they tested this particular bait, SET1, versus the control strain, so the one with the histone. And we have these proteins here that possibly they are part of a complex with SET1. And we have some others here that is strange that they are in this part of the graph because they are not supposed to be like lower expressed. But then what they said, they said, okay, instead of using the control group, let's use the entire background. So let's take, since the background are all the proteins in the yeast, all the proteins that also like between those proteins are also unspecific binders. Unspecific binders should be present in all the experiments. So we don't care which bait was used. The unspecific binders should be always there. And so we can use all these number of unspecific binders for each experiment to calculate a background. So we'll do like something like a mean across all this data that we have. And you can see here, if we increase the number of other experiments used to calculate the background, then we will get more significance because it makes sense. We will have more data. We will have more data to confront with the specific binders. And so with 6 other experiments used as a background, we have this data here, and then we remove these 2 weird proteins. As you can see here also, they are nice. We can say that, for example, with certainty that this CPS50 is a very specific binder. While here before, it was in this area of the graph, and maybe we were not very sure about how trustworthy was this result. But then if we go up to 17, background with 17 other experiments, you see this graph is not big enough to show where these proteins are. So we're really sure now that those are specific binders. Another way to test their result, they calculated the profile plots. Here, if you remember from yesterday, they are the same. And then they checked how much they correlate between each other. So if we expect that if these proteins, for example, the red ones here, form a complex together, then we expect them to have a profile plot that is very similar. But like, for example, let's focus on this one, SDH3. If we will analyze only this experiment, you see, focus only on this gray line here. If you focus only on that one, so only on that experiment with that bait compared to all the background, we may think that also SDH3 could be part of a complex, because it's overexpressed here as it is in all the other experiments. But then, if we then check the profile plot, we see that this protein is present also many other times in many other baits. So we can say the correlation is very low, probably is not a specific binder. So, and as we can see, it ends up in this part here of the graph. And this was another way to be sure that what they found were specific binders or not. So how to represent the results that I show you so far? We have different techniques to do so. We will go through only the edge list and the network during this talk. But there are also others we can just represent with a matrix. You have the line, like as you saw before, we have the line of baits and then the praise, and then we say, okay, let's put a +1 where these two proteins interact. And then this is this image that you can see here, this is a multi-node network, aka also as a hairball. But I mean, it's called that way because it's not a nice name. As you can see, it's a very messy graph. The only thing that you can recognize here are colors, different colors around. But this is also to show you about networks, to be aware that putting too much information may result in a network that doesn't carry any more information, and that doesn't carry information anymore because it's too messy. There are too many stuff to look at, and everyone who's going to look at this image is not going to understand anything. So for this reason, these graphs are called, when they have many, many nodes, so many, many dots in the network, these are called hairballs, just to say it's like a ball of fur that you don't see anything, and you should entangle that to just try to understand what is the meaning of this. But let's go with the first two of this list. So first, we have here the edge list. As you can see, is a very simple and simple table and simple way to represent the binders. We just have a source, a target here. We say, okay, if we put this protein here, then this one will interact with this one, with this other. We can add additional columns. For example, we can add information about the type of interaction that happened between these proteins. You know, it could be, for example, an enzyme and its substrate, or could be an antibody and its antigen, or whatever. We can also add further information about how this interaction was discovered. I will show you a bit later about that. And yeah, that's it. It's very simple way to represent the data. But if we want to create a graph, then we will go for a network. As you can see here, we have this protein that binds with these three others. So we can represent this mode interaction in this way. We have ERBB2 at the center that interacts with these other three proteins. If you can see here, this, I just draw it it has no, I will say, biological meaning what I put here. I'm not sure which kind of interaction happened between these proteins, but this was only to show you that we can carry more information also through the network. We can see that, for example, this interaction is in both senses. So both this protein, whatever you take a bait for ERBB2 or a bait for DLG4, you will find all of them. You will always find the other protein, but maybe in this case, it's not. If you bind then PAK1, you're not going to find this protein because for some reason, because the type of interaction that happens between this protein. So you can add always further information in this graph, changing also the colors of the dots that are called nodes. So remember the node, this part is our protein, and then we have here the edges that are the interaction. So we can change the colors, we can put more information about how this interaction was discovered, how it is, and so forth. Then here I want to show you two networks. I don't know if you're already familiar with them, but one is a good network and the other one is not. And how is it possible to discriminate between a good network and a bad one? Well, the first idea is that usually in biology, the networks tends to have to be more like this. Networks that have a significant a meaning, a biological meaning, tends to be like this. This is because usually you have a protein here that interacts with several other proteins. While in this one, you see there are very few proteins that interact with a lot of other proteins. Usually they just have 3 or 4 interactions. But how to do this in a mathematical way? So we take this node, we take their neighborhoods, and we just count the number of neighborhoods that are present here. So we can say that the node 1 here has 6 neighborhoods, so has degree of 6. And then we go for the others. These will be, all of them will be degree of 1, and this one will have a degree of 2 because it has 2 neighborhoods. And usually in biology, this is what happens. You have one node with a higher degree, and then it drops immediately, the number of degree. So if we plot then, like for example, the degree distribution, we were going to see something like this. And we will call this scale-free, this network. And we'll see, you see, like for example, we have this node, this one, then this other with a higher degree. So they will stay here, and then slowly, not slowly, but quite rapidly, the number of degree decreases. While if we have a random network, this was a network just created with a random number of nodes and edges. Being random, it will follow more normal distribution. So we're going to have not so many nodes with high degree. We're going to have a lot of nodes with like 2 or 3 degree of 2 or 3, and then it will go down again. So when you see that your network, if you calculate all the degree in your network, and you will see that then they have a normal distribution, probably there has been some errors in that network, and probably your network is not so reliable in that case. I mean, maybe everything was done right. It's just a problem that were maybe some errors during the experiment or something. It means that probably it has not a very important scientific value, biological value. But what I showed you before was specific to that experiment. They calculated networks using that experiment, but not all the time you can create a specific experiment. If you want to study interactions between protein, you have the time nor the money to then check every single protein you're interested in to see with what it interacts. As you can see, also it's quite difficult to be sure about which are the interactors of this protein, and you have to prepare a really time-consuming experiment like the one that I showed you before. So fortunately, we have online repositories. Here there are some of them that you can use. I honestly prefer string, but it's just, I find it very nice, the interface, and I like very much the colors that we have. But I don't see any reason why you shouldn't use any one of these other repositories. They're all as good as the others. So just to show you what you will see in these repositories, for example, this is an example for string. I search for this protein here at the center, PRDX1, and then string gives me all the known interactions. The program looks through the repositories on internet and published data, and then we'll find all the possible interactions that has been described so far. As you can see, we have also other information depending on the color, the protein. It will carry then also the structure inside if the structure was described. And then we have a lot of edges here with a lot of different colors. What do they mean? I will show you shortly after. Just before moving to Perseus, I will then open string and just show it to you how to do a very quick search in this database. So now it's time to move to the practical part. As I said, we will repeat this experiment from Kihlauer et al. 2015. We will use Perseus to do so. And you will have already a session in the USB stick that we're going to open now, and sooner I will show you. But in that session, the data preparation was already done. So was already done what we did yesterday. So it's nothing new actually. Okay, just we'll go quickly on string to show you. Here it is. You can see you have a lot of data here. Let's search again for the protein that it was interest. We just say, here we have a lot of species. Also, we can check also for if they have been described, also ancient species of Homo, but let's go for Homo sapiens. Then the program will tell us, okay, there are these proteins. Maybe you were looking for this one because it's an interactor with PDRX1. No, I was interested in PRDX1, so the first one. And here it is. As you can see, it's the same graph as before. But what are the colors? You see here, you can change a lot of stuff here, but I'm not going to, we don't have time, unfortunately, to show everything that you can find on string. But let's focus only on this part, on this legend. We have some known interactions that has been, for example, found with an experiment or through a database search. And then we have predicted interactions as well. And then others, like these are further information. Like, for example, if these two proteins are co-expressed together usually, so they might be interacting, but they also might not. And instead here, for the predicted interactions, these were found through, you see there are different reasons for that. For example, they were close in the genome, so maybe they're part of the same gene, so maybe the same gene creates two proteins, and so maybe these two proteins interact to each other, but we're not sure. So I will say, if you're really looking if you're really interested in a protein and you want to know exactly what are its interactors, I would suggest you to focus on this one, this part, because it will tell you that these are for sure interactions that has been described. And then here you can play around with the settings and do some little statistical analysis as well in string. But yeah, let's move to Perseus now. So you have your USB stick now. If you go, again, software, then you have Perseus version, and let's open it. What we're going to do now is to open the session that you have already in the USB stick. So you go into software, again, Perseus sessions. So remember, we have already the basic tutorial, and then now we go for network. I will just go quickly through what was done here. As you can see, we removed the contaminants. We then This is an interesting thing because we didn't show this in the previous classes. So as you can see from this matrix here, the column name is incredibly long and doesn't carry any information that we are interested in except from the bait that was used and the number of replicates. We have all these parts. I mean, we don't need that in the columns. So we renamed the columns, but we have, as I said before, there are 30 baits here with 4 replicas each. So there are a lot of columns. Renaming each one of them singularly will take too much time. So there is this function that I will show you here. We have it in rearrange. We can rename columns, but here you will do for each column, you have to do that. Otherwise, we can do this. And this will pop up. And here you can write, I remember that all the information that I needed in that column was after final. There was the name of the orbiter up, then was written final, and then the bait information. So here, you just have to write dot and asterisk. And this means that everything that is until what you're going to write here is going to be selected. So we will write final. And everything after this will be replaced with, everything before what we've written will be replaced with something here. But we don't want to replace it with this symbol. Let's do like that. As you can see, this was the previous names. These were the previous names. And here you're going to have the preview of what you want. Here you can also replace with whatever you want, like, yeah. But in this case, we just needed the name of the baits. And now we have only what we need. We clean all the stuff, and then also the different replicas were grouped and everything like what was done yesterday. Then transformation, filter rows based on the values, replace missing value. And so we ended up with this nice matrix that, as you can see here, there are the cleaned column names with their grouping and all the data. There are no missing data at all now. So we can start with our, we can start with the network now. The first thing we're going to do is to go here. You see, this is the Hawaii plots. This was also something that was asked yesterday during the question session. So this is a way to represent all the experiments together, to calculate all the volcano plots and have them all together in this part here. So you can see you don't have to repeat for each experiment the volcano plot you're going to have for all of them. So what we have here, we want here you just put which names you want the protein to have. Gene names is totally fine. Then here we have the grouping. What we're going to analyze the groups across the different groups. So it means we're going to analyze across the different baits. So grouping is fine. Negative control. If you remember, these were the negative controls, the histons. But it was better not to use the negative control, but to use the entire background. So what we will do, we have here another possibility that is complement, and we select complement. In this case, we are not going to do the comparison between just the negative control and the baits, but we're going to do the comparison between baits and all the unspecific binders that were found in all experiments. Here we can decide the number of permutation, then the FDR. Let's keep, these are the information for the volcano plots. Let's keep them as they are. So let's give the PC a bit of more time to load all the volcano plots here. Let's make nicer here the interface so you can see better Okay, now as you can see, we have for all the baits that were used, we have a volcano plot in which we can see here the unspecific binders, and then over here, the specific binders. Let's check, for example, the first one, APC1. So we go here into this table where we have all the results of all the comparison. We then bring to the top this selection. Here you see significant ones. Yes, you have here significant, here significant class B. So it means with all the background where they were compared. Let's just select these proteins. As you can see here, with the FDR, then only these were found significant. So let's select only these. To select different rows, just press Ctrl and then the row that you want. And here we have these specific binders that were specific binders for APC1. Then it makes sense that for APC2 probably is a very similar protein. We have the same specific binders here, just one more. And we don't find them in the other experiments. So it means that they bind only APC1, only that bait. As you can see here in this graph, we can find it, but here was just one of these genes was just an unspecific binder. So it just ended up doing some noise here, but that is what we expected actually. So then we can move on. If you don't like this, like they are too small here, we can select one volcano plot, and here will be enlarged. And you can see also the names, the name of these specific binders. Of course, there is APC1, it binds itself, but we have also other proteins here. And yeah, this is more or less what we can say about these Hawaii plots, because as you can see, there are many volcanoes, so it's called the Hawaii plots here in Perseus. But with this information now, we can create the base for a network. If you remember, we have the edge list, a first representation. Actually, now we were going to create a matrix. So we will have baits on one side, and then the praise on the other side of the matrix. How to do so? We just go into this part of the graph. We have create network from significant interactions. So only the significant interactions will be selected for our network, and that is what we want. Yeah, I know, don't worry. Then, and then we will just leave this information as it is, just leave the default parameters. And now here, as you can see, you have this green slot here, network collection. We have just some information about our network here, but we're not interested in then. What we are interested in is this. Here we have the information about which are the baits, then leave this part. These were just the categorical information for the matrix, don't focus here. But here we have the information about the baits, so the nodes of our network. And if it's a bait, then we will say, okay, yes, so select it, this one as the center. And then we have the edges that will tell us if these proteins interacted with each other. They are the same here and here. So we'll say, okay, APC1 interacts with APC1 as expected. But then we have APC2 interacting with APC2 again, and so forth and so on. And then here we have the other binders. Now that we have this information, we can create the network. So we will just go to network over here. Here you have other functions. As you can see, it's the same. The add here is the same as it was in matrix. The idea is that matrix, you work with matrices, network, then you have the function for networks here, and then the visualization here as it was for matrix. What we will do, since we pulled down the network and we created this matrix, now we're going to pull up. All the basic informations are just fine. We just press OK. Yeah, now you will see. And here it is. So now we represented all the interactions that we saw in the previous volcano plots. As you can see, in the green color, we have the baits that were used, and then in blue, then we have their interactors. Just to give an idea that this makes sense, I mean, we are actually looking what we saw before in the volcano plots. Let's check these two proteins. You can see these two baits, they have a lot of interactors. NUP84. Let's go back to the volcano plots, which were NUP84. Here they are. As you can see, it makes sense. There were a lot of interactors here. Sorry, let's not select this stuff. Then we have NUP84 with even more interactors here. And that is exactly what we're finding right now here. Or let's see before, also because before we talked about APC, let's check here. Let's check the names, CDC, MND. Here they are, all of them. So with this, I will finish then my presentation about the network and interactions.